Lung cancer is one of the deadliest diseases worldwide, especially becoming more common in developing countries. Sadly, fewer than 18% of patients survive 5 years after diagnosis, but recent research has uncovered an intriguing twist. Our lungs, long assumed to be sterile, actually host tiny communities of bacteria, viruses, and other microbes. Imagine your lungs as bustling cities. They come into contact with air from the outside world every second of your life, yet scientists used to think they were totally germ-free. Recently though, we discovered that even healthy lungs have residents. Microbes like bacteria, which include groups called firmicutes, proteobacteria, and bacteroidetes. Some specific examples are Prevotella, Veillonella, and Streptococcus. Other bacteria, including Enterobacteriaceae, Haemophilus, and Ralstonia, also make a home in the lungs. But like any city, problems arise when the balance between good and bad residents is disrupted, a situation called dysbiosis. When beneficial, peaceful microbes decline and aggressive pathogenic ones multiply, the result is ongoing inflammation. These bad neighbors produce substances that irritate lung tissue, causing chronic inflammation, disrupting normal cell functions, and creating conditions that favor cancer. One particularly notorious resident linked to lung cancer is a bacterium known as Chlamydia pneumoniae. Researchers have found that individuals with chronic infections caused by C. pneumonia have around 1.6 times higher risk of developing lung cancer. But how exactly does this tiny bacterium drive such serious illness? Scientists believe inflammation might be the key. When infected by C. pneumoniae, your lungs experience chronic inflammation generating harmful molecules called reactive oxygen species, ROS. These molecules directly damage DNA, increasing mutation rates in lung cells. Normally your body can handle some DNA damage, but chronic inflammation speeds up cell division, increasing the risk that damaged cells survive and transform into cancer. Moreover, C. pneumonia has other tricks up its sleeve. It produces special molecules that suppress your immune system, create persistent inflammation, and even shut down tumor-suppressing mechanisms that usually keep cancer at bay. Remarkably, these bacterial molecules can infiltrate human cells, embedding themselves within vital cellular compartments like mitochondria, the cell's energy factories, the nucleus, the cell's control center, and the endoplasmic reticulum, responsible for protein production. By interfering with these critical organelles, C. pneumoniae disrupts normal cellular functions and further pushes cells toward cancer development. Another intriguing factor is a protein called chlamydial heat shock protein 60, or CHSP60. Even when chlamydia is dormant, quietly hiding in your lungs without causing symptoms, this protein is continuously produced. Acting as a toxin-like molecule, CHSP60 triggers your immune system, leading to chronic inflammation and possibly even autoimmune reactions. It activates certain immune sensors in our bodies such as TLR4, causing cells to release inflammatory molecules, IL-6, TNF-A, Illinoid-1B, that further fuel cancer-promoting inflammation. Additionally, scientists using computer simulations discovered that hundreds of C. pneumonia proteins invade human cells and disrupt their normal processes, especially in mitochondria. By doing so, they prevent cells from dying naturally, a process called apoptosis, and disrupt normal cell division, accelerating the transformation from a normal cell into a cancerous one. In essence, these tiny microbial invaders, once thought harmless or even non-existent, can dramatically tip the balance in your lungs, promoting an environment ripe for cancer. By understanding these hidden microbial communities and how they affect our health, researchers hope to discover new ways to prevent or even treat lung cancer, revealing the microscopic world inside us as both a challenge and an opportunity for future medicine.